안녕하세요, royalib 제이퍼 강사입니다. Today we will look at a database question from the 2021 May exam. So this question focused on mitosis. It gives you a whole blurb about some of the extracts used from avocado and crepe jasmine. And they are basically looking at the effect that avocado extract and crepe jasmine extracts have on mitosis. Now, as you can see, the mitosis comparison is done in mitotic index, which is basically the percentage of cells undergoing mitosis given a certain set. And so here, as you can see, when we increase concentration, we actually can see a decrease in the mitotic index for both cases. And so for avocado, when you increase the concentration of the extract, mitotic index decreases. And it's the same for crepe jasmine. When you have increase in concentration, you also get a decrease in the mitotic index. Now, they start at the same percentage of 8.3, but avocado has a greater effect in decreasing the mitotic index, since it ends up at 2.11% compared to 3.72% of crepe jasmine. The first question asks you to calculate the number of cells undergoing mitosis at the 100 parts per million crepe jasmine extract. And so that would be a total of 3,000 cells. And it gives you the mitotic index of 8%. And so to find the number of cells, we multiply 3,000 by 8%, which is 8 over 100. And that gives us a total of 240 cells. And so that's the number of cells undergoing mitosis at that concentration of crepe jasmine extract. Now the next question asks us to describe the effect of avocado extract on the mitotic index. And so as we previously looked at the data, we see a decrease in the mitotic index with the increase in concentration of the avocado extract. Now for question B, they want us to compare and contrast the effect of increasing extract concentration on the mitotic indices for the two plant extracts. And so here, whenever you get a question that says compare and contrast, they're asking you to write down the similarities and differences. You do need at least one similarity and at least one difference for this question. And so the similarity here would be that increasing concentration of the extracts end up decreasing mitotic indices for both avocado and crepe jasmine. You both start at 8.3. But as the data show, the avocado extract results in a greater decrease. So that's a difference that we're going to discuss there. Now, when you are answering DBQ questions, it's always helpful and beneficial to mention data points. And so in this case, for avocado, we're going to note that it's more effective at lowering the mitotic index because the mitotic index starts at 8.3%, the control, and it's lowered all the way down to 2.11% at the maximum concentration of extract. Whereas for crepe jasmine, you do start at 8.30, so the same baseline. But at the highest concentration of the extract, you end up at 3.72%, so a little lower compared to avocado. However, though, crepe jasmine appears to be more effective in lower concentrations when compared to the avocado extract. Okay, so we're looking at that 1,000 to 2,000 parts per million area there. Okay, and so that's another point that we're going to write. We're going to say crepe jasmine extracts are more effective at lower concentrations of extract compared to avocado. And so we have 3 points here. We have 1 point for the similarities, the second for the differences, and then the third discussing the effectiveness at the lower concentration. Now for the next set of data, they give us the percentage of cells in different stages of mitosis given the different concentrations of the plant extracts. Now, as we can see, when we increase the concentration of the avocado extract, we see an increase in the number of cells in prophase, whereas the number of cells in metaphase, as well as the cells in anaphase and telophase, decreases. A different pattern is observed with crepe jasmine, though. With crepe jasmine, when you increase the concentration of the plant extract, we get a decrease in the number of cells in prophase, but we see an increase in the number of cells in metaphase. Now, the differences in the number of cells in anaphase to telophase is insignificant for the set of data for crepe jasmine. For C, they want us to discuss the effect of avocado extract concentration on the percentage of cells in the different stages of mitosis. So that's the graph at the top. As we can see, and as discussed before when we looked at the data, the increase in concentration of extract results in an increase in the number of cells in prophase, whereas we see the opposite effect on the number of cells in metaphase and anaphase, as the increase in concentration of extract results in a decrease in the number of cells of metaphase, anaphase, and telophase. Now for D, they want us to use both the table that we saw previously and the bar charts, and they want us to deduce whether the extracts actually contain chemicals that block mitosis in the given broad bean root tips. Now, the extracts, as we saw in the given data, do have an effect on mitosis, right? When we look at the table data, we did see that the increase in concentrations of both plant extracts resulted in a decrease in the mitotic indices. Now, with respect to the data given in the bar charts, as we can see and discussed before previously, increasing the avocado concentration results in an increase in the number of cells in prophase. But the opposite is observed with respect to cells in metaphase, anaphase, and telophase, because we see that the number of cells in telophase, anaphase, as well as metaphase decreases. And so what this actually shows us, and what we can deduce from the given data, is that the number of cells in metaphase, anaphase, and telophase decreasing is actually resulting from cells being blocked in prophase. So cells are not being able to transition from prophase into metaphase and anaphase towards completion of mitosis. The next set of data they give us shows us the effect of increasing vinblastine concentration on the percentage of cells in mitosis. We are also shown by the graph on the right the effect of increasing vinblastine concentration on the ratio of cells in anaphase to those in metaphase. And as we can see from the graph on the left, when we increase the concentration of vinblastine, we get an increase in the percentage of cells in mitosis. Now that's in contrast to the graph on the right, because the graph on the right actually shows us an opposite pattern, where when you get an increase in the concentration of vinblastine, you actually see a decrease in the ratio of cells in anaphase to those in metaphase. And so in question E, they ask us to evaluate the hypothesis that vinblastine targets cell in mitosis and prevents the cells from completing the process. And so by looking at the two graphs given, we can actually state that the hypothesis is valid, as we see that increasing its concentration does increase the percentage of cells in mitosis. And so that validates that first part of the hypotheses. However, when you look at the graph on the right, we can see that with increasing concentration of vinblastine, we're actually getting a decrease in the ratio of cells in anaphase to metaphase. And so we can deduce from the data given that perhaps the cells in metaphase are getting stuck there because they cannot transition into anaphase. And so for F, they ask us to suggest a way in which vinblastine could potentially block mitosis. Now, if you reflect back on the processes involved in anaphase, we have shortening of the spindle fibers so that the sister chromatids can be separated and move towards the opposite poles. And so if a cell is stuck in metaphase, then perhaps vinblastine is preventing the separation of the sister chromatids. And so we could talk about perhaps there's prevention of separation of the sister chromatids. Now, the other one that we can mention is perhaps the prevention of shortening of the spindle fibers as well. Then they go on to ask you to discuss one advantage and disadvantage of using plant tissue to investigate drugs intended to treat cancer in humans. Whenever you take a study from one species and you try to apply it to another species, the obvious disadvantage is that cells may not respond the same way. Now, when you look at data obtained from plants, with respect to its application on humans, the major advantage here is that no human is harmed during the investigation. And so there's less risk to humans. And so our advantage would be that there's less harm inflicted on humans. Now, one of the major disadvantages would be that data may not be applicable to human cells. And so human cells may respond differently. That brings us to the end of part 1 on DBQ. And I will hopefully see you all in part 2 where we discuss excretion. Today we will look at a database question focused on excretion. Now, a lot of students have difficulty maneuvering their way through this question. This question is focused on a specific type of turtle which lives in saltwater marshes. Now, turtles can live underwater as well as out of water. They have fully developed lungs as well as kidneys, but they also have microvilli in their mouth to exchange oxygen and urea. And so the hypothesis that's stated here is that they can take oxygen and excrete urea at the same time. And so if you look at the table given, they compare excretion of the nitrogenous compounds of ammonia and urea from the mouth versus by the kidney. For turtles immersed in water versus the ones that have immersed in water. And so you look at turtles submerged in water versus the ones that are out of water. Now, for ammonia, there isn't a significant difference between the excretion of ammonia by the mouth in turtles in water versus those out of water. But when you look at the excretion of ammonia by the kidney, for turtles in water versus out of water, you could actually see that there's a decrease in the amount of ammonia excreted. For urea, a different pattern is shown for both cases, be it for mouth or kidney. You'll notice that the amount of urea excreted actually goes up. And so question A goes on to ask you to deduce whether the excretion of ammonia or urea changes more when a turtle emerges from water. And as we analyzed before, we can see that the change is greater for urea. Now for B, they ask you to compare and contrast. And so we already know immediately that we're looking for at least one similarity and one difference. And they're looking for that difference for the change in urea excretion in the mouth versus for the change in urea excretion in the kidney when the turtle emerges from water. And so we're looking at the bottom column there. Now the similarity is that in both cases, be it by the mouth or the kidney, we are seeing an increase. Now that increase is also the same value. If you subtract 0.9 from 1.56, you'll notice that the change is 0.66, which is also the same change experienced when you go from 0.07 to 0.73. Now the main difference, however, would be the percent change. And the percent change is much greater for the excretion of urea by the kidney because you go from 0.07 to 0.73 versus the increase from 0.9 to 1.56. And so percent change is greater for excretion of urea by the kidney. Now in the next set of data that's given to us, we're shown percentage of saturation of water with dissolved oxygen versus the time. Now as we can see from the data given, when the heads of turtles are dipped in the water, we can see that the percentage saturation of water with oxygen actually decreases. And over time, what are you seeing? You're seeing accumulation of urea in the water as well. And so C1 asks you to describe the trend shown by the graph for the amount of dissolved oxygen in water discharged from the mouth. And we can see that since when water is submerged, oxygen actually decreases with respect to the percentage of saturation of water with oxygen. And that when its head is stuck out of the water, you can see an increase in that percentage of saturation of the water with dissolved oxygen. And so what are we seeing? We're seeing that when the turtle's head is in water, we're seeing a decrease in O2 saturation. But when the turtle's head is out of water, we're seeing an increase in the percent saturation with oxygen. Now, why would such trends be observed? Well, when the turtle's in the water, it can't use its lungs. And yet it still needs oxygen to carry out aerobic respiration. And so what is it doing? It's sticking its head in the water and absorbing the oxygen from the water. And remember, you can't use your lungs underwater as well. And so the only way to access the oxygen needed is by absorption of oxygen from the water. Then they show you the effect of an inhibitor, fluoretin, on the amount of urea excreted from the mouth. And as you can see, compared to the control, there's a drastic decrease of the amount of urea excreted. And so it says, deduce with the reason whether a urea transporter is present in the mouth of P. sinensis. And as we can see from the bar chart here, and the drastic decrease, we can say that urea transporter is indeed present, as in the presence of fluoretin, we all get almost zero urea transport and excretion. Then lastly, what they show you is the output obtained from gel electrophoresis. And as we can see from the output, we can see that the mRNA for the protein is expressed in the tongue and at the roof of the mouth, shown by these bands. And we get no bands here, so there's no expression there. And so it says, outline the additional evidence provided by the gel electrophoresis results shown above. What did we say? You have mRNA bands only present in the roof of the mouth and tongue. The presence of the bands, which is basically the presence of mRNA, show that there's actual gene transcription and expression here at work. You're only getting mRNA because you are actually carrying out transcription, and therefore gene expression is fully active. There was another set of data. Okay, so this is actually the last one. Now in this case, they show us the amount of mRNA expression of urea transporter following the injection of 3 different substances. So they can either inject the turtle with a salt solution, which is isotonic to that of the turtle, they could also inject ammonia or urea. Okay? And what we can see from the bar graph here is that when we inject ammonia, the relative amount of mRNA expression increases the most. And so they ask you to identify the control, and we said the salt solution is isotonic, so therefore our salt solution is the control. And then they ask you to reason why there's a greater expression for the gene after injection with ammonia. Now remember, ammonia is very toxic. And so it can't be stored. It has to be gotten rid of right away. Ammonia, for excretion, is then converted into urea. And so therefore, if you increase the amount of ammonia present, you're going to increase the amount of urea present. And to get rid of this urea, you need to increase the number of urea transporting proteins. It's stated that the salt marshes in which turtles live, the marshes periodically dry up to small pools. And so they ask you what could be a problem caused by this change in the environment, and how then turtle behavior can overcome such a problem. Okay. Now, since you're having pools drying up further and further and further, and marshes drying up, what are you seeing? You're going to see that concentration of urea will become significantly higher. Now, how will turtles behave to this? Well, they'll try to adapt by still dipping their heads into the pools. But diffusion may not be efficient enough. And so now they may have to employ active transport to get rid of the unwanted urea. Okay. Now this is a 3-mark question, so we're talking about concentration of urea increasing in the pools, and to that the turtles will adapt by still dipping their heads into the small pools, but instead of diffusion, they may actively transport using ATP. This brings us to the end of this section on database questions. Thank you for listening.